Hello, everyone. Welcome to our next session. We are happy to have Caio Seguin from Australia, and it's early morning. I know. Thank you so much for accepting our invitation. I'm going to invite Bratislav to do the introduction.
Hello. Hi. Thanks everyone for coming. Hi, Caio. It is really my pleasure to introduce Dr. Caio Seguin from the University of Melbourne. In particular, I'd like to say a big thank you to Caio, not just for keeping up with the delays, but also it is very early for him. And we're very fortunate that he's agreed to give a talk here because Caio is actually an expert on network communication in the brain, has done more than anyone in the field really to understand this link between structure and function and how they're linked through communication models. His work is very exciting and instant kind of reading material for us in the lab. You're in for a treat. And again, thank you so much, Caio, for agreeing to give the talk. Really looking forward to it.
Thanks, Bratislav. Thank you for the introduction. It's my absolute pleasure to be here, and especially speaking in a session with you chairing and Rick presenting and Amy presenting, a lot of people who have inspired a lot of the work that I've done. So I'm really happy. And yeah, sorry if I sound or look a bit tired. I just woke up, and because of that, I missed the previous talks, but hopefully there won't be too much overlap there. So today I'm going to tell you a little bit about my research on modeling network communication in the human connectome. And I would like to start, let's see if this is going to go. Oh yeah, here we go. I would like to start with this really nice figure from a recent review by Bratislav and Suarez and colleagues. So here we show a structural connectivity matrix mapped with diffusion MRI. And on the other side, we show a functional connectivity matrix mapped with correlations between both signals. And the key point here is that only a tiny sliver of the functional connections in this matrix are backed up by underlying structural connections. So the question is, how do we get these structural connections when we have no anatomical connections linking these nodes? So it must be that this functional connectivity is being supported by polysynaptic communication paths. So here, this is an example of what I mean. We have two regions highlighted here in orange. And if we assume that they have a functional connection between them, the question becomes, what is the structural path in the underlying anatomical network that supports communication between them? Here in green and red, I'm highlighting two possible paths that could do this job, but it's very easy to see that there are many, many, many possible paths linking these two regions. So the question becomes, what are the routing and propagation strategies that facilitate communication in the connectome? Can we use tools from network science and graph theory to uncover these principal paths of communication in the brain? So I'm going to tell you a little bit about really a very brief summary of the work that has been done in brain network communication models. So the sort of first big family of models is this idea of information routing, where information going from a source region to a destination region flows along a single communication path. And the second family is this idea of diffusion, that information starting from a source sort of oozes through the network along multiple fronts at the same time. And from these two big families of communication models, a lot of more specific strategies have been proposed to model neurosignaling. So shortest path routing is by far the most traditionally used communication model, and to the point that it's sometimes tacitly assumed that communication always takes place via shortest path routing. And really the advantages of this family is that if communication really flows from a source to a destination via a single path, this is very metabolically frugal, so that is definitely a plus. And this also engenders very fast and reliable communication between two regions. The problem, though, with this family is that it is hard to see how an individual brain region would be able to compute the shortest path to a certain destination. In order to compute shortest paths between two regions, you really need to have knowledge of the whole network. So you need a bird's eye view of the network. In other words, the centralized assumption of knowledge. And on the other hand, we have the diffusion family that is also split up in a lot of different models. So we have work by Bratislav with linear threshold models. We have communicability, which is a very famous model. And we have random walks, and a lot of work has been done on that as well. And they're kind of the other side of the coin, because sending information along multiple network fronts is very inefficient from an energetic point of view. And it also entails a lot of synaptic retransmissions, which could be error prone. But on the other hand, these models work by very simple and biologically plausible mechanisms. So these two families propose, they're kind of laid out on a spectrum from centralized models on one end to decentralized models on the other end. And all of these communication models can be seen to populate different positions in this spectrum. And as you move to the extremes of the spectrum, you have shortest paths with very complex knowledge assumptions, but very efficient communication, to random walk models that are decentralized and they do not have complex knowledge assumptions, but they entail a very energetically expensive way to send signals around. This is really a crass underestimation of the number of the communication models that have been proposed in the literature. It is just to give you an idea of what are the concepts that people are working with in this field. So what can we do with these communication models? The main idea is that once you've decided which communication model from the sort of routing to diffusion spectrum you want to work with, you can compute that from every node pair of the human connectome, which is a sparse matrix, and that will give you an estimate of how efficiently two nodes can communicate. And that will yield a communication matrix. So they allow us to go from a sparse representation of interactions that only capture direct anatomical connections to a sort of a simulated dense matrix of interactions that capture how all node pairs might communicate directly or in a polysynaptic way. And now I want to tell you a little bit about network navigation, which is a model that we have proposed. So the main motivation for network navigation comes from the question, what if we could achieve efficient communication that characterizes shortest path routing, but without the unrealistic assumptions of centralized knowledge? So navigation proposes a way to route information based on local knowledge of distances between nodes. So the distance that you want to use to guide navigation is arbitrary. It can vary depending on your network. But for spatially embedded networks, the Euclidean distance is a good start. So the way it works is a very, very simple rule, is that starting from an initial node, wanting to reach a desired target node, you simply progress to your neighbor that brings you closer in distance to that target. So it's a greedy routing approach. So I'm going to give you an example of how this works. So this is a toy network. It is embedded in a 2D Euclidean space. And we want to send signals from node A to node H. Here in red, I'm showing the shortest paths from A to H. So the shortest path is A, B, E, H, and that takes 3 hops along the network. So how would navigation send information from A to H? So remember, starting from a source node, you always send the signal to your neighbor that brings you closer to the target. So A has 3 nodes, B, C, and D. But D is the one that is closest to H. So navigation would progress to D. From D, we go to F. From F, we go to G. And from G, we go to H. So navigation goes from A to H in 4 hops in the network. A key point here is that navigation is not guaranteed to work. It can get stuck in these loops and never reach the target. So in this example, we want to now go from B to F. The shortest path has 3 hops here. But navigation would actually send the information first to E, because E is the closest neighbor of B to F. So we would go to E, and then from E, we would go to H. But H has 2 neighbors, E and G. But E is closer to F than G. So we would cycle back to E, and we now get stuck in this loop. So this means that navigation has failed to send information from B to H. So we wanted to see if this very, very simple way to send information would actually work in the topology and geometry of brain networks. So in order to test that, we decided to benchmark the efficiency of navigation to that of shortest paths. So we came up with this measure called the efficiency ratio, which is essentially the ratio of the navigation path length to the shortest path length, average across all nodes in the network. So the shortest path length, by definition, sorry, the one here, is smaller than the navigation path length. So this quantity is between 0 and 1. The closer it is to 1, the closer navigation it is at finding efficient paths in the network. So in the 2 examples that we had here, shortest paths had 3 hops in the network, and navigation had 4 hops. So this is an efficiency ratio of 0.75. So in other words, navigation is sending information at 75% of the optimal efficiency. And when navigation fails, we can operationalize that as an infinite navigation path length, and therefore the ratio is 0. So we computed that in the structural connectome. Here on the horizontal axis, we have a group structural network computed at different connection densities. And here in the vertical axis, we have the binary efficiency ratio, so computed in unweighted networks. And we have the weighted efficiency ratio, so computed in weighted networks. And in red, I'm showing the results for the empirical human connectome. And we can see that even for very sparse networks starting at 10% density, navigation is achieving 80% of the efficiency of shortest paths in the binary case, and around 70% of the efficiency of shortest paths in the weighted case. And this is with navigation having no clue of what the weights in the network are. We're just looking at the distances between nodes. And in green and blue, I have two node models in which I rewire these networks. The green one is the rewire network, so a Maslow's mapping, and the blue one is a reposition network. So I'm shuffling the spatial position of nodes in space. So both of these node models lead to marked decreases in navigation efficiency. So this really points to a 3-way relationship between brain network topology, geometry, and communication. And the key point of this paper was to illustrate that with different strategies to send information, we can still achieve very high efficiency given the topology and geometry of the brain, but without the unrealistic knowledge assumptions entailed by shortest path routing. So another really interesting property of communication matrices is that they can be asymmetric. And that's true even for undirected networks. So I'm going to give you an example. Here, this is a toy network. It is undirected, so you can traverse both connections bidirectionally. But if you want to send information from I to J, this is the path that you would follow according to the rules of navigation. But if you want to send information from J to I, this is the path that you would follow, the purple one. So the efficiency of sending information between 2 nodes depends on the direction of flow between them, even in an undirected network. I'm not going to go through the details here, but this is also true in the case of measures based on diffusion, based on random walks. So here we have diffusion efficiency and search information. They are also asymmetric in undirected networks, but shortest paths isn't. So it's really the decentralized character of these measures that leads to the asymmetry. So we wanted to explore okay, we know that these measures are asymmetric, but does this really lead to anything interesting when we look at it in the brain? So here we've projected navigation asymmetry at the regional level onto the cortical surface. So nodes in orange and yellow were nodes for which outgoing paths were more efficient than incoming paths. So we call these the senders. And receiver nodes were nodes for which incoming paths were more efficient than outgoing paths, and we call these the receivers. So a sender node does not mean that it cannot receive information. It is just that it is better at sending, statistically better at sending than it is at receiving. And the nodes that are shown in gray here did not show any statistical difference between their sending and receiving abilities. And what was really cool here is that prominent senders included all the primary sensory regions, so A1, V1, S1, and M1. And prominent receivers included higher order regions such as precuneus, frontal, and prefrontal cortices. We compared these with the sort of unimodal to transmodal gradient defined by Margulies, and we confirmed that unimodal regions are biased towards sending information, and multimodal regions are biased towards receiving information. And we think that this makes sense. So, so the idea is that regions that are sending results just based on how they're positioned in the topology of the connectome might be better wired to quickly progress those sensory stimuli to the rest of the brain, while multimodal regions might be better wired just to integrate this stimuli. And what I think is really important here is that I'm focusing on the results of navigation, but this was true if we looked at search information or if we looked at diffusion asymmetry. So I don't think that this is particular to a certain way in which communication is being modeled. It seems to be really a property, a topological property of how the brain is organized. The next thing that we wanted to see was to see if this asymmetry was going to agree with a functional measure of asymmetry. So to do that, we got resting state fMRI data, and we computed the principal components time series of activity for M functional modules. So you can think this, for instance, as the Yale pacillation. And we actually run spectral DCM on this. And this gave us a matrix for each subject of effective connectivity. So this is now a directed functional matrix that is telling us about putative causal relationships between regions. And we downsampled our communication asymmetry matrix, again, computed from the structure, to the same resolution of functional systems. And the idea is that we wanted to see if the asymmetry that we saw across the structurally derived sort of asymmetry and the functionally derived asymmetry would agree. And this is what we saw. So here in the bottom axis, we have the navigation asymmetry. And here in the vertical axis, we have effective connectivity asymmetry. So we saw a really nice correlation between these two. And this was also true for the other measures, let's say search information. So we thought this result was really cool because it's saying that diffusion MRI networks can actually be used to study asymmetric functional interactions, which is something that, because diffusion MRI cannot resolve fiber directionality, was thought was not possible. But really, the asymmetry is embedded in the topology and how the topology interacts with these different communication models. Another thing that we did was to try to predict human behavior from communication matrices. So what we did is we got a bunch of participants from the Human Connectome project. I think this was around 1,000 participants. And we essentially vectorized their communication matrices as well as their functional connectivity and structural connectivity matrices. And we computed behavioral dimensions from the behavioral data set of the Human Connectome project, and we fed that to a machine learning algorithm. So really, this is a very summary picture of this project. And we can see that here in green, functional connectivity was by far the matrix that gave the best prediction accuracy. So the key point, though, is that structural connectivity here in pink was outperformed by many of the communication matrices that we looked at. So the idea is that if we transform structural connectivity matrices using these communication models into these dense communication matrices, we are improving the predictive utility of the structural connectome. We're not doing as well as FC, but we are closing the gap between the two. And I think this is, in a way, to be expected, because functional connectivity, in a sense, is what these communication matrices are trying to model. And it's reasonable that we're not all the way there yet. So functional connectivity does a better job at predicting behavior. But by modeling these polysynaptic paths, we can start to close the gap. We can start to approximate the behavioral predictive utility of functional connectivity. So I thought this was another nice demonstration on how these communication models can help bridge the gap between structure and function. Okay, so all the work that I've shown was using functional connectivity as a way to sort of validate what we can compute based on structural connectomes. The problem is that I've been measuring functional connectivity using fMRI, and I've been talking about signaling and communication and signal propagation. And fMRI only captures these things at a very slow temporal resolution. So we've been looking at this data set in collaboration with a French group, which is a data set of intracranial EEG. So these are invasive recordings done in patients with epilepsy. And what they did is, because they had such a large number of patients with epilepsy, they could actually collate experiments across many patients, to the extent that electrodes were positioned in such a rich spatial arrangement that they could derive a whole brain matrix of cortical evoked potential amplitudes. So I think this is really exciting, because the idea is that you stimulate node I, and there's an electrode here at node I, and there's an electrode here at node J, and you can actually measure the response at a millisecond scale, which is really what we want to get into, right? This is really what neurosignaling is doing. And the idea is that, can we now model this whole brain matrix of amplitude using network communication models? So this is the project that I've been working on lately, and we have only some really preliminary results. We seem to find that the distance between electrodes is a big deal, and the results really changed based on how far apart the regions are. And it's kind of interesting, because we see that for regions that are really close by, models that are based on ideas of diffusion, such as search information, give better predictions. And as the electrodes become further and further apart, models based on the idea of routing become more predictive of cortical evoked potentials. So the idea is that perhaps there's not only a single mechanism governing communication in the brain, but depending on what the particular node pair that you're looking at is, this mechanism will be different. So if you imagine for nodes that are really close by, or perhaps belong to the same structural module, a simple diffusive strategy would be enough to send information between them. But if you look at nodes that are really far apart and perhaps belong to different functional systems, that is when you would require a long distance routing strategy to be able to integrate information. So we're kind of seeing some preliminary results in that direction, but this is still early days, and I'm excited about this project, and I'm keen to see what happens here. So just to conclude, some final remarks. I hope I conveyed a sense that brain network communication models offer a framework to integrate structure and function. And this is really a complementary framework to a lot of the other ideas that are out there in the field. So there's a lot of talks on control theory here. This is a complementary approach to that. It's a complementary approach to dynamic models of neurodynamics. But it's a very simple approach, and that's really the strength of these models. They're very simple and they're very easy to compute. A key point is that which models most faithfully describe biological neurosignaling remains unknown. So there's a lot of efforts in proposing new models, but I think studies that consider a lot of different models in the same paper to try to see which model gives the best predictions of whatever you're trying to understand, that's still lacking. And I think that will be an important thing to consider if we want to disentangle these different mechanisms of routing information in the brain. And I think to do that, new methods will be really important. Rick's going to talk about edge time series. And I think this is an exciting development that could help us actually track the sort of temporal spread of information in the brain that could help us then relate this back to these ideas of structure. And I think also perhaps moving to different data modalities that can capture neural activity at high spatial temporal resolution, such as intracranial EEG, could also provide a way forward. Yeah, that's it. That's it from me. Thank you.
Wonderful. Thank you so much, Caio. That was a very stimulating talk and very profound, I think. And it has elicited quite a few questions, which we will cover. So we have 4 questions in the question box and 1 in the chat. So I will just read those out as we go along. So Rick asks, have you tested the efficacy of other distance metrics for navigation other than Euclidean distance?
Yes, that is a very good question. The answer is kind of. So when I was first working on this, we tested this idea of kind of a linear combination between structural weights and Euclidean distance. And that, like mixing in a little bit of structural weights to guide the routing process seemed to help in some stages. But intuitively, if you go all the way to structural weights, navigation would not work because you would just get stuck in very high weights connections. I sort of played around with the idea of using gene co-expression data to guide navigation. And if you get genes that are very spatially distributed and kind of follow Euclidean distance, then it works. Which is kind of just saying that you're using Euclidean distance again. But it's interesting because Euclidean distance seems like a really silly and simple measure, but it's actually quite hard to beat navigation using Euclidean distance. Yeah.
Yep. Rick also asks, why does FC perform better than other measures? I guess this is in your network neuroscience paper where you were predicting behavior. Is it because there's simply more variability in functional connectivity than structural connectivity across subjects?
Yeah, that's a good question. The way I see it is because the interactions between regions are what is driving the prediction of behavior. And FC is really capturing, in a data-driven way, these interactions between regions. So the sort of patterns of synchrony, which engender behavior and perception, are captured in a data-driven way by FC. With all the problems that FC measured by fMRI has, it is still capturing it in an empirical way. Whereas communication matrices are a model, and it tries to approximate FC in a sense. So my take on it is because FC is actually the real thing, whereas the communication matrices are still an approximation that doesn't take us all the way there.
Yeah, makes sense. John asks, and I will paraphrase the question a little bit, how in the navigation setup, how would a source node know how far its neighbor is from its eventual destination? How do they compute this other metric space?
Yeah, that's a really good question, John. And that is the I always get this question, is the main criticism to the idea of navigation. And the idea is that you're right, it is still a unrealistic assumption. So here we have to assume that a node knows about the spatial positioning of its neighbors in relation to a desired target. And I can't really tell you what are the biological mechanisms that would allow for that. But the key point of that paper was not really to say that this is what is happening in the brain, was just to illustrate that by using a way to propagate information that entails a lot fewer assumptions than shorter sparse routing, we can still achieve high communication efficiency. So the key idea is that this spatial knowledge is, in terms of information, is a much weaker assumption than knowing the entire network, which is what you need to know to compute shorter sparse between regions.
Yeah. I'm just going to ask a related question, which is not the next one in the list, but it's from Marina, who actually suggested, could it be that the choice of route depends on the present state of the neurons that are nearby? And so the next time the route would change because the neuronal states are changing?
Yeah, absolutely. This is a really good point. I mean, these models are really simple and they abstract away the whole sort of dynamics of what's going on at the nodes or at the systems as a whole at any given point in time. This is purely based on the structure, on the topology of the structural network. So I agree that this approach can only take us a certain part of the way. And I think perhaps hybrid models that would consider topology plus the modeling the dynamics of what's going on in the nodes might be good.
Yep. So then I'm just going to ask one last question here from Matteo. I apologize to the others, but we kind of are trying to get back on schedule. We can continue this offline, or online later. But Matteo asks, does the navigation change if you assume different directions, and how reliable is the undirected case?
So you mean different directions as in directed connections?
I guess so. Matteo, maybe you could clarify in the chat.
Or maybe he means directions in terms of going from node I to J as going from node J to I? Or, oh no, I think he answered yes. Okay. Yeah, so navigation is impacted by directed connections. But we also computed it on track tracing networks from the mouse and the macaque and the fly as well, and it still works. It still works. We also, in the Nature Communication papers, we compared computing navigation on undirected networks with computing navigation in directed networks, and we actually saw that there's a really nice agreement there. So I think that would have an impact, but the strategy still works to route information.
Great. Okay.
I think that's great. Yeah.
Fantastic. So I'm just gonna maybe close the session right here. Apologies for the two questions that are left unanswered, but you can follow with Caio later.
Yes, happy to talk.
Thank you so much, Caio. That was a wonderful talk, and we will see you all in the final session by Rick Metzel.
Thank you. Thanks, Caio.